All right. Hi, everyone. We're going to continue learning the biology. So we're moving into next chapter, which is the dynamic cell. This is chapter number 5. And in this chapter, we're going to talk about the different type of energy. We're going to focus a little bit more on like just how the cell is able to function. So energy is the capacity to do work, right? Like why do we eat? We eat so we have energy so that we can like move around, so we can like study for our exams and everything. And energy, the main source of energy is from the sun, right? And we have two types of forms of energy, potential energy and kinetic energy. So looking at this picture, think about when you're skiing, right? So when you're skiing and you're on top of the hill, you have potential energy. You're not moving yet. You have that potential energy. As you're moving down the slope, that's your kinetic energy, this energy and motion, right? Another place that you can see stored energy is like in the super here. There's all this heat, there's all this, the food itself has all these bonds, and there's a lot of stored energy in that soup. As we consume it, we can take that stored energy, break those bonds, and then we can, from there, get kinetic energy and use that kinetic energy to, for example, fuel us running a marathon, right? So potential and kinetic energy goes back and forth between the two. You know, we have potential energy that can be converted to kinetic energy. There's kinetic energy that can be converted back to potential energy. So it's kind of like this feedback, what back and forth between potential and kinetic energy. And so, as I mentioned, when we think of energy, we tend to think of food we eat, right? And the food we eat has energy in it because, like I mentioned, when we talked about carbohydrates and we talked about protein, that a lot of it is that breaking bonds. Remember, whenever we talk about energy, we're talking about breaking bonds, and by breaking it, we're releasing energy. So, calorie, the way that we measure calorie is the amount of heat energy that's needed for us to raise the temperature of 1 gram of water by 1 Celsius. That's how we measure calorie. So how much calorie does it take to raise that 1 gram of water by 1 Celsius? So you can tell that something has a lot of energy because of how much heat it can release, right? So just think of like when you guys are like out there, you're roasting marshmallows, how quickly that marshmallow just poof into fire. If any of you guys have ever done it, s'mores at fire, I feel like, you know, the marshmallow looks good, and then next thing you know, it's like on fire because there's so much energy stored in that marshmallow because it's just pure sugar. And so sugar has all these bonds in it that just allows us, right? So the amount of calorie that marshmallow has is how much heat energy is released in order to raise the temperature of 1 gram of water by 1 Celsius. And so let me ask you guys, you probably have heard of like when people talk. I know my dad used to always say that eating crab burns more calories than like actually trying to break the crab, getting the meat out. It's more calories than you actually get from actually eating it, right? So think of like the celery you eat. When you chew, you swallow the celery, does that burn more calories than what you actually get of calories? Calories pretty low celery, not calorie, celeries is pretty low calorie to begin with, right? But it's not just the chewing and the swallowing that takes up so much energy to do. It's actually your body digesting the celery because the celery, if you think about when you eat celery, it's so fibrous. They're so and so for us to be able to break all that fibrous tissue into something that our body can actually try to extract energy from. Um, you probably are burning more energy trying to break down that celery and chewing it than you are from the amount of calories that's actually in it. Okay, so going back to kind of energy and how I told you that energy can move back between potential and kinetic energy and go back and forth. This kind of falls into this first law of thermodynamics, which is that energy is neither created or destroyed. So we can convert potential to kinetic and we can convert kinetic back to potential, but we never actually create any new energy, nor do we destroy energy, right? So we just transfer these energy forms back and forth. And so the second law of thermodynamics is that energy cannot be changed from one form to another without a loss of usable energy, which means that we always, no transfer is completely perfect, and we always lose a little bit of energy. And a lot of times this is released as heat. So we can actually harness that heat and use it, but that heat will then be released. And so it's it's always a little bit of energy that's lost. And this leads to an increase of entropy, which is like also disorder. And disorder in our universe is always increasing. We're just always increasing entropy. I tend to think of it like how my house, how no matter how much I clean, it's always messy. Like it just always gets messy. And that's because my house is always increasing in entropy. It's always increasing in disorder, right? Um, so energy can be changed into another form, and that form is often heat, right? We can see that from the sun. The sun, the energy that we are able to, that plants are able to harness in order to convert the energy into food source and for itself, and that later we will consume, is because of that energy that's from the sun being changed into a usable energy source. And so for us in our body, our energy currency is ATP, adenosine triphosphate. And this is a nucleotide. We talked about nucleotide in the last lecture, and how we have ATP and GTP and CTP and TTP. Sorry, I had to think like, what am I missing? So when we think of energy, we can actually get energy from the other nucleotides too, but our body tends to use ATP the most. And this nucleotide, its energy is actually stored in these phosphate bonds right here. So ATP is triphosphate, right? So it has 3 phosphate bonds, and it is actually the energy that it takes to lose that phosphate. So ATP can easily become ADP, and whenever ATP becomes ADP, some of that energy is released. And when it is released, we can take that energy and actually use it. And so the ATP converting to ADP is what drives most reaction in our body. It's what gives us that energy. And so we can think of ATP going to ADP and phosphate as a way to release energy. Converting the other way, ADP and phosphate back to ATP, that actually takes energy for us to be able to do, right? So we tend to think that, and you should know this because it's kind of like the foundation of how we use energy in general, right? So work that we do in our body requires us to have some sort of energy. And the main source of energy, everything from digesting that celery to being able to stand straight or even sit in a chair without slouching, that all takes energy. And so we need energy in order to do all of these day to days. And like our body is always functioning. So there's tons of stuff that's happening in our body that we're not even aware of, that's also using like ATP to harness it. And it's the creatine that kind of helps make ATP. So you can see here, oh, sorry, the creatine turned into creatine phosphate. The creatine phosphate donates that phosphate, right, to make it so that ADP has a phosphate, and that ADP and phosphate would then consume energy in order to make ADP. But then once it's formed, we can later take that ATP and use it elsewhere to actually harness energy to do whatever it is that we need to do. And so the main way that we get energy is either through, well, we can't photosynthesize, but plants and certain algae can use photosynthesis and can take that solar energy and convert that solar energy plus water and carbon dioxide into carbohydrates, right? So whenever we say carbs as far as food, we tend to think of like bread and rice and potatoes, but actually eating that lettuce also has a lot of carbs in it. It's just carbs along with fiber, and fiber is also considered a carbohydrate. But that's what the plants are making. They're doing a photosynthesis and they're synthesizing these carbohydrates, and then we can actually consume those carbohydrates for energy in the sense that our body will convert those to glucose, which will then convert to pyruvate and go through this whole cycle in order to make ATP. The other way that we can make ATP, as I'm kind of alluding to, is cellular respiration. So that's when we take the carbohydrate and we break it down, and this all happens in the mitochondria. We break it down and we actually use that carbohydrate, so we could use those glucose molecules, whatever, to then make ATP in the mitochondria. So mitochondria and the chloroplast is pretty similar. They both make some sort of energy. It's just that one of them is using solar energy in order to make products that have energy, whereas mitochondria is using glucose, mainly glucose, uh, to then convert that to ATP. So mainly um, sugar molecules is kind of what I think of when I think of glucose. So being able to take that sugar molecule and being able to convert it to ATP. And so this allows us to have be able to harness and do a bunch of different reactions. And uh, there's a lot of steps that it takes in both photosynthesis and cellular respiration to take either the sun or to take the glucose molecule and make it into ATP. And we'll start learning about that more a little bit later on. But I'm going to change gears right now and talk about enzyme substrate, because we're going to actually see in our lab how we can actually kind of break things down in order to get energy in a certain way. And we're going to do that by actually looking at enzymes. So enzymes are protein, and these proteins are able to help a reaction happen faster or happen at all. Sometimes there's certain reactions that are thermokinetically, you think that it will happen, but the circumstances just makes it difficult for it to happen, so it doesn't happen. So you need these special proteins to come in and kind of help that reaction. So the substrate is what the enzymes will work on. So that's kind of like on our left-hand side, we have substrate. Those enzymes will then find your substrate, get the reaction to happen, and then you actually end up with products at the end. So here's an example. The enzyme will come and there's some sort of substrate, and the substrate can vary, it can be multiple different molecules. In the case of the lab, it's going to actually be the carbohydrates that's found in potatoes, and then we're actually going to use the enzyme, the catalase enzyme, to kind of break that up. So this enzyme, what it does is it's going to take the carbohydrate, it's going to help make the reaction happen. So you end up with this enzyme substrate. And this is a very specific thing because the enzyme has specific sites on it that allows the substrates to attach to it, right? So not everything is going to just attach to any of these enzymes. That would be chaos if everything just attached to one another and just reactions just happen. So it's very specific as far as which substrate binds to which enzyme. But once the substrate binds to enzyme, you form this enzyme substrate complex. And then once you have that, you actually can then have a reaction happen, and the reaction will lead to the substrate becoming products. So in this example that we're showing here, we're actually taking the sucrose and we're breaking the sucrose via this enzyme into glucose and fructose. And once we have the products ready, then we can release it, and then the enzyme is empty. The enzyme can go and find another substrate to bind to. And this reaction go back and forth. So you can actually take glucose and fructose, use an enzyme, and convert that back to sucrose, for example. It really depends on the reaction. Some of the reactions can go both ways. Not all reactions and enzyme will allow you to go both ways. So it's really like a case by case basis. But just keep that in mind that that's what's happening. Is the enzymes kind of helping catalyze a reaction that might otherwise have a hard time happening and converting those substrates into products. So enzymes can also be inhibited. There's multiple ways that enzymes can be inhibited. You can have another component or another substrate that blocks the enzyme and makes it so that it can't bind the substrate that we're wanting to can't bind onto the enzyme. You can interfere with the actual protein directly itself and make it so that it has a different configuration, so it can't bind. Um, we can actually have another product that binds the enzyme that makes the enzyme again change its shape. So now because it's changed its shape, the substrate, even if it recognizes the shape is kind of weird and it can't bind onto that substrate anymore. So there's multiple different ways in which we can have enzymes not be able to get the reaction to happen with the substrate to the product. So enzymes can be inhibited. So let's kind of review what enzyme does. Again, as I said, the enzyme kind of helps have the reaction happen. So this is what's happening here, is that do you see this red hump right here? That's how much energy it normally would take for the reaction to happen. So in order for us to be able to have that, we have to be able to get over that hump. And sometimes the hump is too steep and we can't get over it. So what we can do is have these enzymes, and the enzymes help lower that low hump. So it helps lower the activation energy that's needed. And because the activation energy is lower, it's easier for enzymes to actually get over that hump because the activation energy is lower. So it still takes the same amount of energy overall to go from product, go from product to reactant. So this portion right here stays the same, reactant and product, but the amount of energy that it takes for us to get over that hump in order to become a product gets lower with enzymes. And so which one of these is not true regarding enzymes? Let's read through and see if we can figure it out. A, they increase the amount of energy required for a reaction to run. B, they speed up reactions. C, they possess an active site. D, they are protein. Or E, enzymes are not used up by reactions. So are they used up by reaction? Yeah, enzymes are being used in a reaction to bind to substrate and making to product, right? And as we mentioned before, enzymes are made of protein. They have that active site because it's the active site that the substrate binds to. And by lowering the activation energy, they actually increase and speed up the reaction. So the answer is A. All right, so let's move. This whole chapter jumps around a lot because it's all just about like the title says, the dynamic of the cell, right? So the cell, we don't want things just going in and out of the cell willy-nilly. So we talked about it, if you remember in lab, before we talked about how the cell is selectively permeable. So certain things can go through back and forth. So we talked about the red blood cell and how water can follow the salt and either go in and out of the red blood cell. So there's certain things that just go in and out of the cells easily, but not everything can go in and out of the cell easily because some things are just too big or some things are non-polar and the cell doesn't want to let it through. So that's why we say the cell membrane is selectively permeable. And there's three ways that substance can't enter the cell. And we want the cells to have substance go in and out, right? Because the cells have to eat. So they need some way to get products into them that they can consume and make energy out of, right? So glucose, for example. But they also need oxygen. They need they need oxygen as well, and they need certain ions. But they also have to get rid of like waste. Like once they use up that oxygen, there's a byproduct of carbon dioxide. So they need to get that carbon dioxide out, right? So they need to be able to get things in and out. And there's three different ways that we can get stuff in and out. There's active transport, active passive transport, and bulk transport. So let's kind of look through each. So the best way to demonstrate passive transport is diffusion and how it doesn't require any energy. So just think in this example, I'm sure you guys have done this, where you have a bowl of water, you add just a drop of dye. What happens over time? Even if you don't do anything, let's say you don't stir or anything, you just drop that drop of water. It will eventually diffuse around and just kind of distribute itself, right? So the net movement of molecule down concentration gradient. So similar to water moving towards salt, all these dye molecules are kind of spreading themselves out to kind of go to go with their concentration gradient. And there's no energy is needed because molecules want to move from higher to lower concentration. So they're just moving across. And this is an example of diffusion because it's set by the cell's own or the substance on itself own concentration gradient. And so it's just moving from a higher to lower position, right? So even when we have two dyes, red and yellow, you can see that it's going to evenly diffuse so that there's red molecules kind of evenly diffuse and a yellow molecule evenly diffuse across the water over time. So if you just let it sit there, it will eventually just diffuse across. Okay, so diffusion is the first one where you just have a substance that just diffuses across the lipid membrane, the lipid bilayer pretty easily. Passive transport is when they're able to move through, and it's just driven by the concentration gradient. So we don't need energy to be added. It's just if there's it's just think back to the whole salt and osmosis. If there's high number of salt in one location versus the other, the water is just going to kind of move. But like, let's say the salt is able to move back and forth. Similar, the salt is then going to move from its high concentration to its low concentration. And it does take energy because it wants to do that. So that's passive transport. So in a cell membrane, passive transport is going to require some sort of channel, protein channel. And then you have active transport. So in active transport, you actually use energy to move the liquid, the substance. So let's say you have a lot of salt, a salt ion, like let's just say potassium. You have a lot of potassium on one side than the other, but you want to create what we call a potential gradient. And so you actually want to force it to go from low to high. Remember, cells that ions tend to want to go from high to low. So if you want to force it to go from low to high, that requires energy. So it's going to use energy, typically it's ATP, and it's going to push that potassium against its concentration gradient. So against its concentration gradient means that it's going to go from its low concentration to high concentration. Normally they want to go from high to low, but here we're going to force it to go from low to high, and that takes energy, and we're going to actually use energy, and that energy is typically in the form of ATP. There's also other ways that our cells can take things in and out of the cell that we call bulk transport, and that's when we form these vesicles. So with bulk transport, what's happening is that rather than just one small ion being moved in and out, we want to move much more and bigger amount of substances. So we're going to actually take the membrane and we're going to kind of wrap around whatever it is that we want to bring in, for example. We're going to wrap around it and then it kind of pinches off. So imagine when you are taking a piece of bread and you're just pinching it off. That's kind of what's happening. Like you're just kind of, you have your membrane, you're just pinching the membrane into a smaller piece, and then that piece is then taken into the cell. The reverse can happen too, where you have a small little piece and you're going to actually push it back onto the membrane, so it kind of merges into the membrane, and then as it's merging, the material can get pushed out. So you can have endocytosis, which is bringing into the cell, or exocytosis when you're pushing it out of the cell. So let's really look at passive transport. So if we look at this picture, how many more glucose will cross the membrane if passive transport is the only force at work? So if we look at this picture and we kind of see, there's three on one side, one on one side. If we only have passive working, we tend to want to even things out, right? So you can easily kind of see what can we do? How much do molecules do we have to move so that there's even number on the outside and inside? We probably have to move one more, right? One more glucose in order to actually get it so that it's even. We'll talk about these um, bonus questions later. Let's move on to the other ones. Do read it, see if it's in your book if you can find it. But you should kind of know what type of transport this is based on whether or not you use energy or not. And you should know if you didn't have a membrane, what would you call this? I mean, not membrane, sorry. If you didn't have a transport channel, because you can see this blob right here, this gray blob, that's a channel, right? So the channel is helping kind of a passage for the glucose to move through. And that's kind of neat because glucose is kind of big. But imagine if glucose could go across without that channel protein, what type of, what would that be called? Okay, so let's visit active transport. Remember, we mentioned that active transport, again, uses energy, right? Diffusion is just follows its concentration gradient. Passive also follows this concentration gradient, but we need that protein channel. And then you have active transport, which goes against its concentration gradient. And you also need some kind of channel to help move it. And you need energy. All right, exo and endocytosis, we've talked about this before. I will link the videos onto Canvas so you can actually watch them. But basically, like we mentioned before, this is this is taking in a bunch of molecules into it, which is endocytosis, or molecules that are inside. Let's say we make some proteins and we really want to push those proteins out of the cells after we make it. We would then put them in these vesicles and then we push attach to the plasma membrane and then push it out. And that's exocytosis, right? It's exiting, so it's exocytosis.
This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane.
I'm going to post these two links for YouTube just because I'm afraid that's going to play a random ad. So I'll post those two. If you have any questions, again, put it in the discussion. I hope that this kind of gives you an overview of how dynamic our cells are and all the different things that's happening in our cells. Our cells are amazing. It's amazing that we're able to do all this stuff and we're not even aware. We're not even aware that things are going in and out of our cells and all that stuff. We're just sitting here living our life, but like just next time you eat like a hamburger or like you're eating, you're licking your ice cream cone and you're consuming that food, just think of like what happens to that food in our body. Like once we go, we eat it, it goes down our body into our belly and it gets digested and it gets uptake. Like what do you think is happening? Like why do we have to consume energy? So I hope you guys enjoy this lecture. If you have any questions, please let me know and I will see you lab. All right, bye guys. Bye.